50,000 years ago, a child died in a cave deep in Siberia's Altai Mountains. All that survived, a splinter of finger bone weighing less than a paperclip, too small to identify by sight, too damaged to classify by shape. But ancient DNA sealed inside would expose something unprecedented, the first direct proof that two lost human species had a child together. High in Siberia's Altai Mountains, Denisova Cave opens onto a landscape shaped by ice, wind, and time. The cave's entrance looks out over a sweep of steppe and larch, a setting as stark now as it was 50,000 years ago. In the summer of 2012, a team led by Anatoly Derevianko pressed deeper into the cave's east chamber, sifting through ancient sediments layer by layer. Every handful of earth passed through a mesh no wider than 0.5 millimeter, designed to catch even the tiniest traces of the past. Amid the dust and splinters, a field assistant noticed something unusual, a fragment no larger than a child's fingernail, brownish, fragile, and half buried in fine silt. It measured just 24.7 millimeters in length and weighed 1.68 grams, lighter than a coin. The team logged it as DCA-227, recording its exact depth, over 2 meters below the cave floor, and noting its proximity to Levallois stone tools and the bones of mammoth and cave lion. The fragment's surface showed pitting and tiny scratches, evidence of its long journey through freezing soil and perhaps even the digestive tract of a scavenger. Derevianko paused over the find, recognizing its potential despite its unremarkable appearance. The fragment was photographed, bagged, and sealed for further study. In the field diary, he described the moment. No larger than a thumb tip, but too human to ignore. The team's decision was clear. This piece would not be discarded or overlooked. Instead, it would become the center of an investigation that would soon challenge the limits of what bones alone can reveal. The question lingered: who did this tiny fragment belong to, and what secrets might it still hold, sealed within its ancient structure? Attempts to classify the fragment began with the tools of classical osteology. Researchers measured, photographed, and scrutinized the piece under magnification, searching for the landmarks that usually distinguish Neanderthal, Denisovan, or modern human bones. But the surface was marred by pitting and faint scratches, likely left by the jaws and acids of a hyena. The fragment's ends were broken, obliterating the growth plates and joint surfaces that might have given away its origin. With less than 50 milligrams available for study, even the most careful metric analysis could not separate this bone from the thousands catalogued in comparative collections. Turning to protein analysis, the team applied ZooMS, collagen fingerprinting that can sometimes sort ancient bones by species. The result was frustratingly broad. The collagen signature matched that of a generic hominin, offering no resolution beyond the human family. The fragment remained an enigma, its identity masked by time, taphonomy, and the limitations of traditional science. Dating, at least, offered some clarity. 3 independent methods, optically stimulated luminescence, uranium series, and electron spin resonance, converged on a window between 48,000 and 52,000 years ago. Dr. Katerina Duka, who led the dating effort, expressed confidence in this range, noting the agreement across techniques and the secure context of Layer 12. Yet knowing when the individual lived did nothing to answer who they were. The analytical path had reached a dead end. Only by turning to the molecular level could the mystery of the tiny bone fragment hope to be solved. Inside the Max Planck Institute's ancient DNA laboratory, the bone fragment from Denisova Cave entered a world defined by caution and precision. Every step took place under strict contamination controls, positive air pressure, HEPA filtration, and ultraviolet sterilization. Before any bone powder touched a test tube, surfaces and tools were treated to remove stray DNA. Researchers wore full-body suits, double gloves, and masks, limiting every possible source of modern contamination. Svante Parbo, a pioneer in the field, supervised the process. The team drilled into the dense interior of the bone, collecting less than 50 milligrams of powder, barely enough for a pinch of salt. To maximize DNA recovery, they added carrier RNA, a helper molecule that binds and stabilizes the fragile genetic fragments released from the ancient mineral matrix. This step proved essential for samples this small, ensuring that even the shortest DNA pieces could be captured and analyzed. A partial uracil DNA glycosylase, or half UDG, treatment followed. This clever protocol removed most of the damaged bases that accumulate in ancient DNA, but left just enough of the original chemical scars to authenticate the sample's age. The extracted DNA was then bound to silica columns and washed through a series of buffers, a process refined for ultra-low input material. Before enrichment, only about 0.3% of the DNA in the extract matched ancient human sequences. After targeted capture, using custom probes designed to fish out hominin DNA, the proportions surged to 45%. This dramatic jump meant that, for the first time, a genome could be built from a bone fragment that would have been dismissed as scientifically hopeless just a decade earlier. Every safeguard, from cleanroom design to molecular tricks, was about one thing: ensuring that the genetic signals belonged to the ancient individual and not to the scientists themselves. Kay Prüfer sat at her workstation, eyes fixed on the first results streaming in from the sequencer. The mitochondrial DNA, inherited from mother to child, placed the bone squarely within the Neanderthal family, almost a genetic signature. Yet as she moved to the nuclear DNA, the data began to tell a different story. Instead of clustering with Neanderthal or modern human genomes, the nuclear sequences formed their own branch, distinct and unmistakable. This was not Neanderthal nor Homo sapiens, it was something else entirely. Comparisons with other ancient genomes revealed a divergence dating back around 640,000 years. The nuclear D pointed to a population as old as the Neanderthals, but genetically separate, a group that had walked Eurasia for hundreds of thousands of years, leaving behind almost no trace except for a few scattered bones and teeth. The team gave them a name, Denisovans, after the cave that had guarded their secrets for so long. But the contradiction between the Neanderthal-like mitochondrial signature and the Denisovan nuclear genome could not be ignored. Standard models of inheritance could not explain this split. Kay Prüfer and her colleagues ran the analyses again and again, each time confronted by the same paradox. The evidence suggested something remarkable: an individual whose ancestry crossed the boundaries between human groups once thought to be isolated from each other. The Denisovan lineage, once just a shadow in the genetic record, now stood as a third branch of the human family tree, and the unresolved contradiction in the DNA hinted at an even more extraordinary story waiting to be uncovered. Prüfer leaned in as the genome data arrived, each line of code translating millions of years of ancestry into numbers. The first wave of statistical tests, D-statistics, designed to detect ancient admixture, delivered an unmistakable result. Exactly half of Denisova Girl's nuclear DNA matched Neanderthal sequences, the other half, Denisovan. No trace of modern human contamination clouded the signal. Rigorous controls set the contamination rate below 1%. To further validate the finding, the team scrutinized the DNA's chemical scars. 15% of the terminal bases showed the characteristic damage patterns of ancient molecules, confirming the genetic material's authenticity. Prüfer plotted the ancestry proportions using QP graph, a tool that models evolutionary relationships. The graph produced a strikingly simple picture: a hybrid child positioned at the intersection of two distinct branches, one Neanderthal, one Denisovan. This was not a remote cousin, not the product of gradual mixing over generations, but the direct offspring of two different human groups. For the first time, the evidence was clear and quantitative. A first-generation hybrid, her genome split down the middle. The implications reached far beyond a single bone fragment, opening the door to a new understanding of how human lineages once met, mingled, and created something entirely new. Genetic evidence now reveals that Denisova Girl was not an isolated case, but part of a much larger pattern. Across Eurasia, ancient DNA shows a web of interbreeding among Denisovans, Neanderthals, and early modern humans. Segments of Denisovan DNA, sometimes as much as 6%, are still found in the genomes of people from Asia and Oceania today. Even some Neanderthals carried traces of Denisovan ancestry, up to 2%, demonstrating that gene flow moved in both directions. One remarkable example is the EPAS1 gene, which helps modern Tibetans thrive at high altitude and can be traced back to Denisovan ancestors. These discoveries come from the analysis of tiny amounts of bone, sometimes just 10 milligrams, using advanced sequencing techniques that recover genetic material once thought lost forever. Archaeological finds, like overlapping stone toolkits in cave layers, support the genetic evidence for contact zones and shared technologies. Svante Pääbo describes this as a complete rethinking of human evolution. Not a simple tree, but a tangled network where lineages met, mingled, and left lasting marks on the human story. Today, our DNA still carries traces of Neanderthal and Denisovan ancestors. Every genome sequenced adds another piece to the puzzle, blurring the lines between ancient species. The past lives within us, reminding us that the story of humanity is one of connection, not separation. What will your DNA reveal?